So, good morning to everybody, and welcome to the first conference that is specifically focused on single-cell proteomics. It's a conference, a gathering of pioneers, both established ones and aspiring pioneers. Among the established ones, we are going to hear from Ralph Weislader on his long efforts and continuing efforts on using very high-resolution molecular imaging for ever-improving disease diagnosis. And Steve Maccaro will tell us about efforts in his group that have resulted in developing one of the most powerful methods for measuring messenger RNAs in single cells. And while this is single-cell proteomics conference, I think many of us have our sights set much more broadly on being able to quantify all of the important molecules in a cell and combining proteomic and transcriptomic efforts. And particularly when it comes to the method that Steve and others have developed, the drop-seq method for sequencing, I think there are a lot of aspects from which we can learn, such as the extremely high throughput of quantifying the transcriptomes of very, very many cells, which is what has really powered applications to important biological questions. And then tomorrow, John Yates will tell us in the morning how he helped establish the current field of modern mass-spec proteomics by first conceptualizing and then demonstrating approaches to analyzing and interpreting mass spectra, being able to assign a sequence to all the M over Z's that we measure, because if there's no sequence connection to biology, they're not really all that informative. We all use these methods on a daily basis. In my group, we mostly use them as implemented by the MaxQuant platform, developed by Jorgen Cox, sitting over there. And I'm very much hoping that we can entice Jorgen to incorporate a lot of other single-cell proteomics algorithms into that platform and make it even more powerful algorithms that we are developing specifically suited and for single-cell proteomics and others, the whole community. And of course, we'll also hear many exciting pioneering efforts by colleagues who are shaping the emerging field of single-cell mass spectrometry. I think it's a tremendously exciting time for that field. There is a ferment of lots of different diverse ideas, and I like it that way, because when there are a lot of different ideas, there is the potential for synergism, and there is a higher probability that we find what really works. And I think that there is a lot of value in being able to get together and discuss how they all fit together and what should be our standards for comparison and interpretation. What should be the standards to say that we have identified a molecule from single-cell mass spec data? What should be the standard for quantitation? Should it be the same as the standards that we have had for samples comprised of millions of cells, or do we need to rethink that? And there is so much to discuss that I have intentionally left a lot of time in the program for discussions and questions, and I would like to encourage everybody to ask questions during the talks, in between the talks. We have long breaks for lunch and coffee. Again, I hope that this will be time for fertile discussions. And at the end of tomorrow, we are going to have a 1-hour period dedicated specifically for discussing and summarizing our thoughts and ideas from the 2 days. And before I turn to the science, I just want to briefly cover a few logistical aspects. The first one is we have been fortunate to be well supported by our sponsors sitting at the back and provide very good meals for breakfast, coffee breaks, lunch, dinners, and so on. The lunch is going to be in this building in room 440. We can all go together so you don't get lost. And dinner is going to be across the railroad tracks. We just cross Ruggles Station. And we'll go to the faculty club where we'll have catered dinner for tonight. Other than that, you will not need to go to many other places of the campus, even though a walk might be a good thing to do. It's a beautiful campus, especially during this time of the year. The other logistical aspect is recording the talks. So all of the talks will be recorded by a professional video operator. And if any of you, the speakers, would like not to be recorded, of course, you should let us know, and your talk will not be recorded, certainly not shared online. Otherwise, I will give you a very short form where you can sign that if you agree that your talk may be uploaded online, which is what I would like to do, if you agree. And on the side of recording the talks in the audio video, one more thing is for the speakers to be able to get higher quality images on the recordings. The video operators would like to have your slides before each session. So if you can just bring your presentation on a USB stick to the video operator, that will be great. Otherwise, you can either use my laptop for presentation or you can use yours. It's entirely up to you. Well, so those were the logistics. And now I'm going to switch and talk a little bit about what enticed me to try to develop methods for single-cell proteomics and what is it that I want to use them for. So about 2 decades ago, fluorescent proteins were a novelty. They allowed for the first time to measure the levels of endogenous proteins in single cells at reasonably high temporal resolution and throughput. And when people started doing that, they found that even among very, very similar cells, such as bacterial cells that came from the same cell and that grew in very homogeneous environment, there were differences in their proteomes. That came as a big surprise to many people and gave rise to a whole field of studying these fluctuations in gene expression. And here we're talking about a clonal population of very, very similar cells, all growing in the same homogeneous environment. Now, if you go to something like a human tissue or a tissue from any multicellular organism, you're going to find that there are many specialized cells. And those cells with different functions have different proteomes and different transcriptomes. And recently, we have begun to learn a lot more about those differences, thanks to methods that have been developed to measure the levels of lots of messenger RNAs in single cells. And this is the type of data underpinning this display of pancreatic human cells. Essentially, the experimenters measured a few hundred to a few thousand messenger RNAs in a few thousand single cells, and they projected these high-dimensional data into just the two-dimensional space of t-SNE, which is similar to PCA. Now, here's an even more impressive and more inspiring example of doing that comes from Steve McCarroll's lab. They were able to quantify the transcriptomes of 50,000 single cells. It's pretty impressive. And those were single cells coming from mouse retina. And based on the transcriptomes, they're able to identify all these different subpopulations of single cells. So that's pretty cool. But why do proteomics? Why measure proteins in addition to transcriptomes? It seems like they're doing a really amazing job. We're doing that? Well, so the transcriptome is very useful, obviously, to classify all of these cells, but it gives an incomplete description. Just to give a couple of anecdotal examples, there can be important membrane ion channels that are present at the protein level, but they're long-lived. They were synthesized a while ago, and they're not present at the mRNA level. On the other end of the spectrum, there could be messenger RNAs that are simply not translated, or proteins that are being translated but very quickly degraded. That is the primary mode of regulation. And in fact, we have evidence that a lot of that is happening. Because when we compare the levels of mRNAs and proteins that have been measured in the same samples, they differ quite a bit in a very special and misleading way. a way that has misled a lot of people. So do they correlate? How accurate are messenger RNAs if used as surrogates for protein levels? In one way, they're very accurate. If we wanted to know which are the most abundant proteins in different cell types and tissues in human, mRNA measurements does almost as well as protein measurements. Ribosomal proteins, tubulin, histones, they're the most abundant proteins in almost any cell, apart from red blood cells and some other special types. But most of the cells that build our bodies have the same set of very abundant proteins, the same set of very lowly abundant proteins. So if we just array together measured protein levels, and these data come from the draft maps of the human proteome efforts, we can get fairly good prediction of the overall abundance for differences in the abundances of different genes. But then if we ask for any one gene in particular, for any one protein, how well we can predict the differences in the levels of that protein across different cell types or tissues, then our ability to do this prediction is much, much more limited. And this type of dependence, where when you condition on a subset of the data, you get the opposite dependence to what you get from averaging across everything, is well known in statistics for many decades as being Simpson's paradox. And this is exactly what we found to be the case for the most part in terms of predicting protein levels from mRNA levels. And these data really underpin the need to also measure protein levels in addition to messenger RNA levels for characterizing single cells and obtaining meaningful measurements. So I'm inspired by that picture. And that's what I want to be able to do with single-cell proteomics. I would love to be able to quantify the proteomes of tens of thousands, maybe 50,000 single cells. Now, when it comes to post-translational modifications and signaling in single cells, mRNA data are even less informative. And that's important because currently, the largest number of ongoing efforts to develop pharmaceuticals is centered around targeting kinases. Similarly, efforts in my laboratory to improve rational differentiation protocols to make different cell types very much focus on trying to affect their signaling mechanisms, which we cannot very well infer from transcriptomics. And that's why we would like to measure not just the proteins, but also their post-translational modifications in single cells. So why is it hard? Now, for at least 5 years, sequencing the transcriptomes of single cells have been growing and approaching becoming a standard method used across almost every major university, every major company. Proteins should be easier to quantify in some ways. Certainly, if we look in terms of abundance, the typical mammalian cell, these data come from fibroblasts grown in culture, but they're really quite representative of almost all mammalians, of almost all cells making up your bodies. The typical cell contains only about 17 molecules from the genes with median abundance, while there are 50,000 protein molecules. So there are a lot more protein molecules, and that's a good thing. It's a good thing, especially because high-throughput methods, such as sequencing and mass-spec proteomics, don't measure every molecule. They sample. We take only a subset of the molecules and we count them. We measure how many we have. And every time when you sample, you have to worry about sampling error, sometimes more, sometimes less. But the problem is similar to picking blue and red balls from a bucket. So if I had a bucket of blue and red balls and gave some to Ezra to pick, and he picked 2 blue balls and 1 red ball, all of you would have the intuition that we cannot accurately estimate the ratio between blue and red balls. He picked only 3. So that is the sampling problem, and that problem exactly fits the Poisson distribution, can be accurately described and characterized mathematically. And by using this mathematical model, very simple, very well understood, we can estimate how severe is that problem for RNA sequencing and for protein quantification. Now, for RNA sequencing, if we try to do this estimate for the median messenger RNA, using about 20% efficiency of reverse transcription of capture, which is on the high end, fairly standard for the best methods, then we can derive that we'll have at least 50% error in our estimates of the relative abundance of messenger RNA, even if everything is absolutely perfect, everything else. So this is not assuming any issues with sample preparation, not assuming any problem with the Illumina sequencers or whatever method is used, just because we sampled a small number of molecules from another subset. And for proteomics, the numbers look a lot better. We are much less efficient. We're going to talk more about that. We face other problems. But because we start from a much, much larger pool of molecules, even if we have lower efficiency, like 0.2% or 2%, the relative error in estimating protein levels would be much, much smaller. And I'm going to show you data later in my slides that we are much closer to this 2% or higher efficiency of being able to deliver the proteome to the instrument than to the other number. So, I already alluded, one of the major problems is delivering the proteins to the instrument if you start from a single cell. And that turns out to be a very difficult thing. And we not only want to deliver them, we want to deliver enough of them. Because if I had a mass spec instrument with perfect sensitivity so that I identify every single ion that goes in, I assign a sequence, that will be a detection device. It will not be a quantitation device. If we are counting single ions, we have to be counting hundreds of them so that we can obtain reasonable quantitation. So we not only want to deliver them, but we want to deliver them in as large amount as possible. And that means we shouldn't lose them before they get there. How we don't lose them? We should try to reduce the volume of sample preparation. And this is something that many of you, including my laboratory, has worked on, and we have new results to share with you on that. But we are very far away from what the transcriptomics community has done. The dropseq method uses droplets that are only a few nanoliters. Our best methods are hundreds of nanoliters. So there is quite a bit of space for improvement there. Now, the 50,000 cells that I want to quantify, I'm never going to quantify them if my students have to pipette them. I have been blessed with wonderful students, but doing sample prep for 50,000 cells just not something I want to subject them to or believe that a human can do very accurately. So we absolutely need more automation in one way or another. And again, this is something that we have done some work on. And for the aficionados of you who are familiar with mass spectrometry, you know that there cannot be a high number of identifications, robust high-throughput mass spectrometry without good separation before the instrument. So chromatography, our ability to separate the molecules is essential. And the more we can improve the chromatography, the narrower are the solution peaks, the more ions we can sample per unit time, and therefore for the same throughput, the better quantitation we can get. So that's certainly an important dimension along which we can improve the methods. The other one is, if we hit a wall, or if we don't want to improve more the chromatography, for the same chromatography, we can sample more ions simply by allowing the instruments to accumulate them much longer. And considering that we currently sample only a small fraction of the elution peak in virtually all methods that are widely used, we can gain some sensitivity and some throughput that way. There is certainly opportunity for instrument improvements. In particular, the ability to collect multiple ions simultaneously and inject them serially is something that I like very much. And Bruker has already stepped in and developed hardware for that. And Thermo is working and trying to do that. So I think that's a very welcome development. This is probably my favorite one of the most important aspects if we want to get to many, many thousands of single cells quantified. Because current mass spec instruments, like the one I have in my lab, they're marvels of engineering, and they're very expensive. I don't know how to make them an order of magnitude cheaper, or two, which is what I would need if I have to spend a lot of instrument time for each cell. So the only way around that is to quantify very many cells at each run. We need to somehow barcode them and label them. And that is generally harder than with transcriptomics. Now, with transcriptomics, there is this unfortunate trade-off that it's easy to make practically unlimited number of barcodes, but when you have a lot of barcodes, then the amount of data and quality of data that you get per cell decreases for the same number of sequence reads. In proteomics, that's not the case. If we could come up with 1,000 or 10,000 or 1,000,000 barcodes, the quality of data is not going to be affected. Well, maybe for 100 million, we'll get into that range, but it's not going to be affected much more, but it's so much harder to make those barcodes. The good thing is that very many bright colleagues, including some of you in the audience, are actively working on making those barcodes, and I'm very confident that we can get to at least 100, 500 plex. And considering that a single run on the instrument can be as short as an hour, with 100 or 500 plex, we can easily get into the numbers of tens of thousands of single-cell proteomes quantified per week per instrument, and that's the realm that I begin to like. That's the realm and the point at which I think we can begin doing very exciting biology and answering questions. So all of these things that I mentioned can be improved with different emphasis. We can either emphasize the absolute best performance that we get for the chromatography. We make a nano LC that is amazing, but you can only run it once or twice before you have to spend a week to service it. Or, alternatively, one can try to push for the best chromatography that is reasonably robust, so that one can run it for a few weeks before one has to service it. And I think these two approaches, they're not mutually exclusive. They can happen together. In my laboratory, we work on both of them. We can both try to make the best Ferrari and the best Ford T model. But my primary emphasis for my lab is on the T model. I really want to have the T model that is low maintenance, that everybody can use and reproduce independently across the world. I think that will be a wonderful thing to get to, and that will allow for the entire community to be able to obtain much more resources for making better Ferraris. Once we have a very solid proof of principle that we can indeed deliver high throughput, high quality single cell data, not the best one that is potentially possible with the technology, but data that are very useful to answer key important biological questions, then there is much more justification to try to push that further and improve it. And usually, the really successful Ferrari parts end up in consumer cars. That's the way the car industry works, and I suspect that something similar can happen here. What used to be a Ferrari part eventually might become a T model part. So one of the challenges with doing single-cell mass spectrometry is that mass spec instruments are so darn sensitive to all sorts of ions, including all these contaminant ions that are incredibly abundant all around us. Before I started doing mass spectrometry, I didn't know how many ions are floating around the air everywhere. It's amazing. So we need to have clean samples that we deliver to the instrument, and there are two ways of doing it. One is one has to be extremely good at removing the lysis buffer. Whatever chemicals we use to lyse cells, as is conventional to use, we should be able to remove them while losing as little as possible from the material. And I confess, timidity. I didn't know how to do that. I wasn't brave enough. So I tried to do the opposite. I tried to focus on being able to prepare samples that don't need to be cleaned up. And one way that existed, Alexander Ivanov, among others, have used previously, to lyse cells without using chemicals is to hit them with a lot of vibrations, to use focused acoustic sonication. There is an exorbitantly expensive machine, I don't want to tell you how much it costs, that can focus energy into a small glass tube and lyse cells in pure water. Lysing cells in pure water is great because when you don't have anything to clean. This thing is very, very low throughput. I would have never gotten useful data from that if it wasn't for Ezra Levy. He did heroic work with that machine to process hundreds and hundreds of single cells that I couldn't have done. So after that experience, it was clear to me that focused acoustic sonication, at least in its current reincarnation, is not the way to go to get to the 50,000 cells. So we focused on developing another method. We mostly means Harrison Specht, a graduate student in my lab, that again lyses cells in pure water and can do this in parallel for hundreds of cells very, very quickly, much more reproducibly, in much, much smaller volume than what was previously possible with Quavaris. And this is something that I have reserved the privilege and honor for Harrison to tell you himself tomorrow. And I'll tell you a little bit about results using this method, but he will present the full development. And very soon, maybe next week or so, you'll be able to delve in all the details in a forthcoming preprint on the method. But I just want to show here a little bit more quantitatively what is the state of art in terms of volumes of sample preparation, where we are and where we can go. So here on the top, we have the normal volume of what we would prepare a sample from 100 million cells, or 10 million, a million cells. We would use about a mil. And then with this Quavaris instrument, the focused acoustic sonication, we used a few microliters, which was the minimum volume we could get away. Now with our new method, the MPOP, we can go down to a microliter or even less. This is what we have done. We have gone down to a microliter, but we can go further down. And the lowest volume, to my knowledge, is actually NPOTS that was recently published and developed. But this is still hundreds of nanoliters. And this is what Steve Makarov is doing here, and others. So we have 2 orders of magnitude, more than 2 orders of magnitude to go down compared to what our currently best volume is. And I think we can do that. We just have to figure out a way how to do it. And the more we go down, the fewer losses we're going to have, and the more economic our sample prep is going to get, because whatever volume we add of whatever reagents we need, that will be less, it will be cheaper. So I'll remind you briefly, most of you are familiar with our approach to quantifying proteins in single cells, but I'll just very briefly remind you how we do that. This is what we did a year ago. So we started by isolating a single cell into a glass tube. Ezra did that. And lysing this cell with the focused acoustic sonication, 1 cell at a time, a painful procedure. And then we digest with trypsin, label with TMT, and mix the samples. Now, the small glass volumes was a way to minimize losses during the sample preparation. But probably the most essential aspect of all this working with relatively standard components was using the carrier cells. So in addition to labeling the single cells here, we also processed 200 cells that once merged with the single cells after the point of merger, they allowed us to reduce losses on the chromatography because many of the peptides that ended up sticking to the chromatography by mass action, simply by probability, are likely to be from the carrier cells. And they also helped us critically with sequence identification. Because to be able to quantify a peptide, we need a single ion that we really know came from that peptide. But to be able to identify the sequence, we need more ions. And the carrier cells provided those ions for identification. And now the new version of doing that, for which it is so new that I don't actually have a very good graphic for it. So I'm going to use the old graphic. So we have replaced this laborious sonication with MPOP that Harrison will tell you all about. And then instead of doing this in tubes, we do it in plates. Because remember, I want to do thousands of cells. A few cells, not interested. And I'm going to tell you about results from having prepared a sample that looks a lot like a single cell sample, except that we have put 100 times more material. So what would be a single cell channel, we put 100 cells. And what would be a carrier channel, we put 5,000 cells. Now, the reason why we did that is because we wanted to have a standard sample that then we can dilute into 100 different dilutions and be sure that those dilutions are the same, that we don't have genuine single cell variability. And if we can remove that factor, then we can focus exclusively on what are the capabilities of the current instruments, the current LC? Can we really do that? It gives us a very clean way to evaluate the entire system of separating the peptides and quantifying them away from whatever might be problematic in sample prep. And because our sample is so clean, we didn't have to clean it up. We didn't even have to use a trapping column. We did a lot of direct injections on a system that David Perlman, sitting over there in the back, set up in my lab. And we did lots of direct injections, one after another. And they all looked virtually the same. I'm showing here the total ion current aficionados know that. So I was very delighted by that we can actually have a sample prepared without cleanup that is clean enough to be injected via direct injection, therefore no losses. And that this thing worked day after day, week after week, without having to replace anything. And then the first thing to consider is, if we just inject 1% of the sample that I told you about, is the signal going to be larger than the noise? Because the way we prepared it, in the single cell channel, we cannot possibly have more than one cell material. It can be less than that, lost from dilution, but it's not more than that. Can we get signal? And here I'm plotting the results from a single set run for 60 minutes, a single scope set, where we have 2 carrier channels. They're different cell types having 50 cells, and then we have a bunch of channels corresponding to single cell material. And what we can see consistently is that the signal that we get here, it's normalized. So I have divided each peptide, have removed divided it by the median intensity across all the channels. So you can see that the channels that have single cells have substantially more signal than the channel that was left empty. And the carrier channels have about as much more as we would expect them to have. About. They're not exactly. And they have more to say about that. Now, there are 2 aspects to it. One is technological, which is the way the Orbitrap works. Whether we can do a comparison between 2 ions that differ by an order of magnitude or more. And there is something iffy about that I don't fully understand. The good thing about it is that I don't need to understand it entirely. Because we do the single cell quantitation, we never take a ratio between the carriers and the single cells. We only take ratios between the carriers or between the single cells or material with similar abundance. And the other thing is, this is by far the smaller aspect, the instrument, the Orbitrap, the technological. The biggest problem that we've had, and we've had a lot of problems, I'm showing you good data, I'm not showing you bad data, we've had plenty of that. The biggest sort of problems that we have come from co-isolation contaminants and cross-labeling. Now, with the procedure that we have developed, it's very simple. It gives us 2 orders of magnitude boost. But we are very susceptible to co-isolation problems. If there are any contaminants in the single-cell channels that are not present in the carrier, we might have co-isolation and not even know about that. Or not be reflected in the overall spectrum. So we have to work extra hard to find it out. So I'm not going to go into all the nitty-gritty technical details of how we do that. But we've seen plenty of evidence of these 2 factors, either problems with the sample prep or with co-isolation, having some contaminants that disproportionately affect the single cells because there's less peptides there that can affect the signal. And therefore, we have built a pipeline to specifically control for that, and I'll have more to say about it a little bit later in a bit. Now, thanks to that system that David Perlman has built, we got really sharp elution peaks, and therefore our MS2 spectra were remarkably pure. So this is the PIF value, precursor ion fraction, computed by the MaxQuant software that we use. Essentially, more than 98% of the signal in most of our ions come from the main ion. There is very little co-isolation. But all of that doesn't tell you if we can do quantitation. So how can we know that we can do quantitation? There are There are more than one ways to evaluate that I'm going to show you, and I think this should be a major question of discussion for all of us. We shouldn't just take it for granted that because bulk proteomics is quantitative, we can just do the same thing, and single-cell proteomics will be as quantitative. So benchmarking, rigorous benchmarking, I think should be one of the important aspects that this community establishes. So the way the particular piece of data that I'm going to show you here, the one way to compare it, is we can estimate the relative level for each peptide or protein between the different cell types that we used. In our case, we used 2 cancer blood cell types. They're quite small, about 10 microns in diameter each. And from bulk measurements, bulk mass spec measurements, we can estimate the relative abundance of different proteins in those cell types. They differ by about an order of magnitude, and this is done by TMT labeling. And then we can compare these relative abundances to the relative abundances estimated in the single cells. And I was quite gratified to see this result. To me, this is a formal proof that we are delivering a very substantial fraction of the ions to the instrument. Because if we were just doing detection, there's no way to get this level of quantitation. If we just had 0.1% of the peptides being delivered to the instrument, because of the sampling noise, the Poisson distribution that I talked about, we would not be able to get such a good agreement between the bulk and the single cells. But I spent a lot of time talking about throughput, and now I'm showing you a couple of single cell channels. So we did do many hundreds of single cells of doing those injections, at least of the diluted sample. And this is what I'm showing you here. So we had a few hundred Q937 cells, a few hundred Jurkat cells that were barcoded within scope MS sets, and those sets were run one after another. So over the period of just a couple of days, we could quantify hundreds of single cell channels within this scope. And I was very happy to see that the cell type separates perfectly by the first principal component. If we only try to separate 2 cell types, we shouldn't need more than 1 component. 1 component is enough. And that is what we see here. And then there is a bit of a separation of a smaller component, which is quite interesting, between the single cells and the carrier channels that we have. So the carrier channels cluster by type with the single cells. That is the first order result, principal component 1. The difference on the y-axis here, that tells you that carrier channels are still a little bit different. And the main difference for that is actually they're less noisy. They give you a little bit higher quality data. But that's secondary. The main thing, the main take-home lesson is with this procedure, only if we can do the sample prep well, we can quantify hundreds of single-cell proteomes in just a day or 2. And we can do this with fairly high quantitative precision. Now, what do I mean quantify? What is the proteome coverage? How many proteins do we get? Well, my view of that is that what is useful to know is not number of peptides or proteins quantified in a single run. That's a good technical benchmark. But the really important thing from the standpoint of using this data in a meaningful way for diagnosis, using it to answer biological questions, we need number of proteins quantified across many single cells. And here what we see is that as we increase the number of cells, we quantify more and more peptides and proteins across more cells. And this data are just using, this is at 1%, at pp less than, this is less than 1% false discovery rate. It is posterior error probability for the aficionados less than 10 to the negative 2. And only using the spectra. But why should we be using only the spectra? I showed you that our chromatography is remarkably consistent. And that would be an informative feature to use. And this is what we have tried to do with a student in my group, Albert Chen, has very, very ably developed a method to globally align the retention times of all of these hundreds of different experiments and use the retention time within a rigorous Bayesian framework to to use that additional evidence, the retention time, in boosting our confidence in PSM assignment, in spectral to sequence matching, either increasing the confidence or decreasing the confidence. And you can see, this is what I showed you before, just this stuff here. So we are comparing the confidence just based on the spectra, the probability of error, and based on the spectral plus retention time. So the highly confident identifications become more confident. But importantly, we get double and triple the number of assignments that previously we couldn't assign with high confidence, and now we can. And this is nothing new, using retention time to boost sequence identification. What is really unusual in this scenario is that because we had so many runs on exactly the same system that more or less contained the same protein material because the carrier is the same, we had less than 3-second deviation in retention time between runs. And the more accurate our estimates of retention time, the more statistical information you gain, the more power we have in identifying the sequences. So we get a lot more power with this method compared to just standard applications of retention time. And Albert has a poster sitting over there, so you can go ask him if you want to learn more about what we have done there. And I specifically want to talk to Jorgen about it, how we can incorporate this within either MaxQuant or the Perseus platforms. All right. Now we are moving to probably the most important aspect of what I want to talk about. We cannot just assume that we can apply the tools that we have for 1 million cells, and they will work with single cells. We certainly have to rethink sequence identification and peptide and protein quantification. We have to be very careful. I have seen it in my lab over and over again, that when things work well, we can get good data, but there are a lot of ways in which they don't work well, and then we get crap. And we should know when we get crap, because we shouldn't trust it. So with the sequence identification, the only thing that I'm going to say at this point is that I think that using the retention time information is a major avenue that can help. But I like very much some existing approaches, such as match between the runs. But I would like to have estimated confidence in addition. I would like to use the MS2 spectra. The MS1 spectra, using it together with the retention time is OK. But I feel much more confident, at least in my work, in using both information at MS1 and MS2 spectra and the retention time, and this is what we have done. This is what I like in terms of the identifications, being able to assign a confidence for each sequence. Now, in the quantitation is something that is that I'm going to focus more, starting with a quiz for you. So here I have plotted 2 proteomes. Total peptide levels estimated from relatively bulk samples. And my question for you is, are these similar? They have a correlation of 0.95. You don't have to answer out loud, but you need to have an answer in your head. Are they similar? There are 2 different cell types. Quite different cell types. So these are Jurkat cells, and these are U937 cells. They have been labeled with Silac. So these are light, have light isotopes. These have heavy. And they actually differ quite a lot. There are peptides and proteins that have more than an order of magnitude difference in abundance between these 2 cell types that are highly reproducible. So what I have done here is I just take the ratios between corresponding peptides. Instead of plotting them on this log-log space, which has many orders of magnitude and that compress the relative differences, I just plot the relative differences into a 2 order of magnitude space. And what we can see is that these relative differences are remarkably consistent from Silac quantitation. And this type of plot is just no good for being able to evaluate whether there are differences in the relative levels between the proteins and the peptides shown here. Because 2-fold difference you cannot see here. you actually can barely see tenfold difference because they're, what, 4 or 5 orders of magnitude. They're just being squished. And this is one of the caveats that I think we as a community should actively try to avoid. We cannot just plot this kind of log-log plots in 5 orders of magnitude as an estimate for relative quantitation, because just about any cell type that I put in here, any cell that I take from your body, if I just quantify the peptides, put it on the y-axis, you're going to get astoundingly high correlation. If you dig into the supplementary informations of the human proteome map papers, you're going to find that too, that if you correlate the proteomes of just about any tissue, they correlate highly. Erythrocytes are an example. You can probably find a few other examples that would deviate, but most of the cells would show this. And if we want to benchmark relative quantitation, we should zero in onto relative quantitation. This, by the way, is, again, the same Simpson paradox that statisticians have described long time ago. And nonetheless, many people fall prey to that fallacy. Now, in terms of benchmarks, my favorite benchmark to use is external standard for the levels of single proteins. I think it's wonderful if we have independent methods to measure the relative abundances of proteins in single cells and then use those as a benchmark to compare to mass spec measurements. Though I say it's my favorite and I have a confession to make. I needed reviewers urging to do that. Even though it's a great thing to do, it's very rigorous, it's not the easiest thing to do because generally getting accurate quantitation of proteins in single cells is hard. There aren't many methods out there that can allow you to get really high quality information that you can rely upon. In this case, we used fluorescent proteins that we could measure by a fact sorter. I think it's very important, especially at the beginning, to have more of these comparisons, of having independent external validation. They're not going to be proteome-wide because of the existing limitations, but they're useful. And then in the absence, in addition to those, in addition to external validation, simply comparing the relative quantitation from bulk samples using well-established methods, cases where we really know work from bulk proteomics, as our benchmark, and comparing those to the results from single cells is something that I think is relatively easy and should be more widely used, rather than these 5 orders of magnitude log-log plots that don't tell us much, other than that the material looked like a human cell. And the other thing that, the reason why I show here this PCA analysis again is because I think it's very useful whenever possible on PCA plots to include bulk material, channels or material that we know the proteome is going to project into the correct space, so that now when you look at this plot, you know that I'm not just showing a few clusters of a few hundred cells that happen to separate maybe because we lysed one of them, we couldn't lyse the other, but they cluster together with the corresponding cell type. You know that they cluster based on validated relative quantitation. That's me. For more than a year, I have been doing this. Now, I told you that experiments can be rather variable. There are lots of ions all around us. If I just cough, I coughed a minute ago, I spew up way more ions than all the single cells that Harrison has prepared this week. And that's a problem because all of that causes trouble. And for about a year, when things didn't work, when I knew the data are not quantitative, I would spend a day or two analyzing the data and figuring out why it didn't work. And I was so busy analyzing that data that I didn't develop a more efficient way of doing it. I was doing exactly what this guy is doing. Closed his eyes, he doesn't see the wheels here. And more recently, I changed the wheels. So what did I do? So we built an automated QC pipeline that reads all of the beautiful, useful information that MaxQuant outputs. My students and I are intimately familiar with all of the tables that MaxQuant outputs. We've spent a lot of high-quality time. We know them by name. They have nicknames, too. And it's incredibly useful information because we can immediately tell, in most cases, just by running my automated QC pipeline, we can see what went wrong. Was it incomplete injection? Was it apex? We failed to sample the elution peaks at the apex. Did we have contaminants? Were these +2, +1 contaminants? So it was remarkably useful. And I truly feel like that guy, having spent, it's humiliating, but I've done a year of this. And I'm much, much happier now that we have this pipeline. And a lot of the credit for that really goes to the way MaxQuant, being open source, outputs a lot of intermediary information, not just the peptide levels in quantitation, but outputs all of the other ions that are very important. The other thing to say is that I love open source. So I'm very happy in looking at ways of incorporating what we have done here, what we have developed, what I mentioned in terms of using retention time in our specific context within Bayesian framework, using the QC pipeline, using the other software that we're developing. I'm very eager to find an efficient way to share this with the community, whether they're part of existing platforms, other servers, or other ways. I think that can very much help the community, especially as more of us do the same. So, going back to all the opportunities. There are a lot of different opportunities that require a lot of different expertise to improve. And what is so good about that is that in front of me, there is a group of 60 or so people who have expertise for any of these things. And there are more people out there who already have the existing expertise. So I think it's going to take a community effort organized with venues like this to figure out what is the most efficient way to work on these opportunities, because many of those offer orders of magnitude improvements. And while I regret for not having spoken much today about biological questions and using these data, I truly believe that if we are able to quantify thousands of proteins accurately across many thousands of single cells, these data can be absolutely transformative for biology, for many of the questions that are dear to my heart, both in the clinic in terms of diagnosis, but also in basic research, in terms of figuring out ribosome specialization, synapses, and their proteomes in translation, and in terms of being able to do very accurate modeling, leveraging these data, because as powerful and wonderful RNA sequencing is, it has this terrible sampling problem that you cannot avoid unless you get every single molecule, simply because RNA is less abundant. With proteomics, I strongly believe that we can get far more quantitative accurate data because we don't have the sampling problem, the Poisson distribution. I obviously didn't do all of the things that I described. This is the wonderful group of people, not all of them. I don't have all of them on this picture, unfortunately, but in fact, a very important person is missing. Bogdan sitting over there. Next to him is David Perlman. Two very important people missing from that picture. But this is Ezra Levy, without whom I would not be doing single-cell proteomics. He was a freshman undergraduate without any research experience who came to my lab, and he made the Cuvaris thing possible, and the cell lysis and all the other things. My experience was incredibly synergistic. My first foray, by training, I'm a systems biologist, and I like mass spectrometry, but for me, developing single-cell proteomics was this very brave idea that I came up with starting my group at Northeastern that I just wanted to try. And if it wasn't for people such as Ezra and Bogdan Budnik who embraced it and who had the complementary expertise, I would be doing other things, probably interesting, but probably not having that potential for improvement. I'm just super jazzed by the possibility of gaining many, many orders of magnitude improvement in our current single-cell methods and being able to use this fruitfully to biology. Here's Harrison. You're going to see him around today, and he's going to give a talk. Gray, who has very ably started a project of his own in single-cell proteomics. Tony is the guy on whom I now rely for keeping the front end of the mass spec as functional as what David and Bogdan could do before. The original team and stage 2 with more reinforcements. And all of this, of course, is made possible by the generous support of NIH. Any questions? So currently, the largest that they've done is a few thousand single cells, which hasn't been truly single cells. It has been done with this diluted lysate. So we make a master, and then we run 1% of it. But I see no limitation of being able to accumulate larger and larger cells. And I expect no difference in terms of doing this with individually picked and sliced cells. So concretely, over 2,000. I think we can do over 20,000 or more, or 200,000, as long as we have the data. There's no limitation to being able to put them together. Absolutely, absolutely. Surprisingly little. So one thing that I didn't talk about is that our experimental design includes reference channels. So we take material that is mixed from the different samples that we'd like to analyze, lyse it in bulk with MPOP, and then we dilute it to a single cell or 2-cell level, and we put it across all the channels. And I have seen much, much, much less batch effects compared to what I have seen in bulk data doing something similar. I think that the reason for that is really here. Because, at least at the moment of what we have done using shotgun approach, of course one can do other things, one can do targeted, but if we use shotgun approach, we mostly quantify the most highly abundant proteins, and those don't get affected as much by co-isolation with other proteins, because less abundant proteins don't affect as much the levels of the more highly abundant ones. And because we have such high degree of spectral purity, co-isolation is not an issue, as big an issue in our data, which I think is usually the biggest problem with concatenating many different TMT sets run separately. So the answer is, what the data that I showed here, for example, on the PCA separation, so here I have a few thousand cells in each cloud. And you can see that by relative quantitation, they all separate perfectly with the carrier channels. So there's no problem of being able to, I have not seen a problem in being able to concatenate our different trends.
Actually, question to this part as well. So,
Jorgen, just a minute for the microphone, because, yeah.
Yeah, question to this PCA plot. So I'm wondering how much of the separation between bulk and single cell is maybe due to imputation, and how much is due to the actual signal? I mean, is this including imputed?
No, so it's clearly not due to imputation. Basically, this PCA plot is a two-dimensional representation of a giant correlation matrix. Now, if you look at this correlation matrix, you very cleanly see the squares of very high positive correlations, the squares of negative correlations. And the thing that is striking is that the correlations between the single cells, they're on the order of 0.95. The correlations between the carriers is 0.92. So it's just stronger correlations. But even if I don't do any imputation.
And if you look at the loadings, I mean, surely in the cell type direction, it will be the cell type markers or whatever. And in the other direction, it's the low abundant signals? Or what do you see in the loadings if you look into this direction? Like what are the responsible proteins?
So generally, the more abundant proteins have less noise, but it's not a very strong dependence. It's not nearly as strong as for transcriptomics. For transcriptomics, it's almost entirely driven by abundance. not entirely, but mostly. In our case, it also depends a lot on co-isolation issues and this and that. So it is correlated to abundance, the level of noise that we see, but not super strongly. And again, I should emphasize this is relatively small. Most of the variance is here.
So how do you do the calibration or normalization of like the protein abundance data? Do you do calibration across different TMT channels?
Across different TMT channels. So we only use ratios when we have a reference in our set for each peptide, the levels that are measured across the different single cells or single cell channels, we just divide those by the level measured in the reference channel. And because the reference channel is identical across all of the runs, then we get, when we concatenate them, we get relative quantitation, which has the same denominator in every single set, and we just put it in one giant matrix.
First of all, thank you very much for your very inspiring presentation, fantastic presentation. But do you think it's time to call in the devil's advocate? So I remember my mentor, Fred McLafferty, I spent some time in Cornell. He was extremely enthusiastic before the results were obtained, but once they were obtained, he would become extremely skeptical and paranoid, looking for any evidence for artifacts or errors of any kind. So to me, this is arguably the most important slide in your whole presentation, and to me it looks deeply suspicious.
Okay.
Because the principal component analysis, the principal component is the signal that is the difference between the cell types, and if you add noise, then this difference should statistically be reduced, and eventually if noise is dominating, then you should get a point at 0, right? The principal component. So it looks like you have no noise in your single cell data compared to the bulk data, and I cannot explain that.
Well, what I can tell you is that if you look at the correlation matrix, the correlations between the bulk cells are a little bit higher than between the single cells. They're not a lot higher. And some of that extra variance is being captured by the second component. There is some spread here that you can see that spread is very small between the single cells. But that's certainly the decomposition of the matrix. Oh, by the way, I should say for everybody, even though we will post all of this as a preprint very soon, but the data, the raw data are already available. So I tweeted a link on Twitter. I'll make all of the data available on my website. So you're very much welcome to take them, analyze them, and see what you get. This is the decomposition of the covariance matrix.
But where is your empty channel?
Oh, the empty channels are removed. They're not shown here.
Why not?
They're mostly empty. They mostly have zeros, so I cannot do PCA without doing imputation. I'll have to exclude these peptides. How would they do it?
Great talk. Thank you. Indeed, very inspiring. I wanted to check with you, how do you make sure that you can eliminate any possible biases introduced at the stages of sample preparation and sample procurement? Do you separate your cells in different cell ways? Do you process them simultaneously? Do you randomize sample preparation? Are there any precautions?
Yeah. So that's a very important question. I think that sample preparation tends to be the hardest part where a lot of the artifacts may come from, either in terms of contamination or biases. In terms of PCA validation, what Harrison is going to describe tomorrow, what we have done, is to completely randomize the positions on the plate when we do validation. So if we just want to demonstrate that we can cleanly separate a few different cell types, we randomize them on the plate together with the empty wells, and then we put them together. Some of the sets contain only one cell type, others would contain only the other cell type. That's what we do. In general, I think that's a very important concern to have. It's actually one of the things that really impressed me very, very positively when I spoke with Ralph the very first time is how concerned he was about collecting cells and not being affected by those issues. I think when it comes to clinical samples and to actual biological experiments, that will be even more important. In the case of these proof of principle experiments of separation, it's relatively easy to solve the problem by randomization. But in general, as we move forward and begin to work on more important, interesting biological systems, I think we have to be very, very careful of how we treat the cells.
And currently you're processing the samples manually, so the timing element
No, it's automatic. It's by fact sorting. I did not mention that. Harrison will talk in more details about it. I'm a very big proponent of automation. I want my students to be designing experiments and doing data analysis rather than pipetting. And the other thing is, as good as they are, robots tend to be more consistent.
All right, good.
Thank you, nice talk. So my question is basically cell biology oriented, since you are working with mitotically active cells that might be in different stages of a cell cycle. I would like to ask if you would expect to see different proteomic profiles of cells in different stages of a cell cycle if you synchronize the cells somehow, or yeah, so the question is there any footprint of the different stages of the cell cycle in your data?
We should certainly be able to see that and separate cells based on it when we do the single cell preparations. In this particular experiment, no, because remember that we started with 100 cells in each well and then we diluted them to a single cell level, so we already averaged the cell division cycle. But in general, in the kind of data that Harrison will present tomorrow and the kind of data that we're generating with single cells, I absolutely expect to be able to see differences due to the cell division cycle and to be able to assign each cell to a different phase.
Yeah, I basically would like to comment on that separation, that Roman's question about why it's that good. Yeah, I agree it's that good. I really like it as well. I think in this particular case, all the noise that we have in this area been kind of out of normalized because you're looking for the carrier channel and the single cell that had been prepared together. That's why in this case, noise been actually automatically kind of mostly removed from this picture. That's how it looks. That's artificially well. But in real simple lab where there will be single cell, not just a dilution of 100 cells, there will be much more than that noise, but still the separation will be pretty much, you know, good.
No, the thing for me, what I would love Roman to do is download the raw data. I'll give you the link. Analyze them, and I would love to see in your pipeline what you reproduce, what you think of them. I think that is what we need as a community. I think at this point we need a lot of independent reproductions of different people and then merging together and having this discussion. And for that, I'm very, very happy that the data are online. You can just download the raw files and see what you get out of them. I think that's really the best way to clarify exactly what is what.
And I want Jorgen to do the same.
Great. Well, I did very poor job as keeping, looking at the time, but I'm a little bit over time, not too much.